Translation. Synthesis of or the formation of polypeptide chain in accordance to genetic information present on the mRNA. That is called translation. So, translation is the process of the formation of polypeptide chain or the polymerization of amino acids in accordance to genetic information present on mRNA, or decoding of the genetic information present on the mRNA, that is called translation. In both prokaryotes and eukaryotes, this translation occurs in cytoplasm. In case of the prokaryotes, transcription and translation both occurs in cytoplasm, but in case of the eukaryotes, transcription occurs in nucleus, but the translation that occurs in cytoplasm. So, in this transcription, different requirements are needed, enzymes are needed, energy also required. Requirement for translation. Requirements for translation. Ribosomes, mRNA, transfer RNA, enzymes, energy in the form of ATP and GTP, activators, amino acids, all they are required for this translation. This translation process occurs on the surface of ribosomes. Surface of ribosomes. That is, the protein synthesis or the formation of polypeptide chain occur on the surface of ribosome. Scientists who first observed that ribosomes are involved in protein synthesis, jaw mechanic. Scientists who first observed that protein synthesis occurs on the surface of ribosome, not inside the ribosome. The person is jaw mechanic. Ribosomes, first one, these are the smallest subunits, smallest cell organelles. Amembranous smallest cell organelles, generally the size ranges 20 to 23 nanometers. Each ribosome contains the two subunits, smaller one and a larger one. In case of prokaryotes, smaller subunit is the 30S and the larger subunit is the 50S, and it is the 70S type of ribosomes. And in case of the eukaryotes, smaller subunit is the 40S and the bigger subunit is the 60S, the 80S. Two subunits unite at the time of protein synthesis only. Any type, either 30S or the 50S in case of prokaryotes or in case of the eukaryotes, the two subunits unite at the time of protein synthesis only, or during the time of translation only. After protein synthesis or after translation completed, the two subunits separate. Union or the separation of the two subunits depend on magnesium ion concentration. The union or the separation of the two subunits depend on magnesium ion concentration. When magnesium ion concentration is more than 0.1 molar, the two subunits unite. When it is less than 0.1 molar magnesium ion concentration, the two subunits separate. So, during the protein synthesis, magnesium ion concentration slightly increases in the cytosol. During protein synthesis, ion concentration increase in the cytoplasm, that is magnesium ion concentration increases. Structures of mRNA and the structure of tRNA already we completed. Enzymes, amino acid activating enzyme, amino acid activating enzymes, and enzyme that is involved in the formation of the peptide bond, they are required. Energy in the form of ATP and GTP. Activators, these are the proteinaceous activators, initiating factors, elongation factors, and releasing factors. These are the different factors comes under the activators. And amino acids, 20 types of amino acids involved in protein synthesis. Now the procedure. First, this translation completes in 4 steps. Translation completes in 4 steps. 1. Activation of amino acid. Translation completes in 4 steps. Activation of amino acid and transfer to the tRNA. Initiation of polypeptide chain. Elongation of polypeptide chain. Termination of polypeptide chain. By these 4 steps, translation completed or polypeptide chain synthesizes. First step, activation of amino acids and finally transfer to the tRNA. Before protein synthesis, both amino acids and ribosomes are in inactive condition. To participate in protein synthesis, they must be activated. For the activation of amino acid required a specific enzyme and energy in the form of ATP. Activation of amino acid plus energy source in the form of ATP and enzyme. Name of the enzyme. Amino acyl synthetase. This amino acyl synthetase enzyme contains 2 binding sites. It is the enzyme. It contains 2 binding sites, 1 and 2. At first binding site, it attaches to the amino acid, and the second binding site it attaches to the tRNA. So when amino acid combines with the ATP and enzyme, that is amino acyl synthetase, a complex is formed. Amino acyl adenylate enzyme complex. Plus 2-PI, PPI, pyrophosphate, amino acyl adenylate enzyme complex. For the activation of each amino acid, 1 ATP required. For the activation of amino acid, each amino acid required 1 ATP. So in a polypeptide chain, if 100 amino acids present, 100 ATP utilized. So number of ATP required, that is equal to number of amino acids. And this amino acyl synthetase is specific to particular amino acid. Here the 20 types of amino acids involved in protein synthesis. So total types of amino acyl synthetases required 20 types. Total types of amino acyl synthetase enzymes present 20 types, that is equal to the types of amino acids. This activated amino acid. Now this amino acyl adenylate complex enzyme complex combines with tRNA. After that, amino acid shifted to the tRNA. AMP release tRNA with amino acid also liberated. Enzyme liberated, AMP liberated, and tRNA with amino acid also liberated. Now the tRNA with amino acid called charged. tRNA or amino acyl tRNA. That means here enzyme plus AMP plus tRNA with amino acid. Amino acyl synthetase consists two binding site. At one binding site, amino acid attaches. To the another binding site, tRNA attaches. Amino acid shifted to the tRNA at the binding site. When amino acid attached to tRNA, tRNA liberated, AMP liberated, enzyme separated. So the three structures separated. Now the tRNA with amino acid called charged tRNA, also called amino acyl tRNA complex. Suppose it is the first tRNA or initiator tRNA that reaches to the initiating codon AUG. The anti-codon is UAC that carry amino acid methionine. So initiator tRNA that reaches to the initiator codon called that carries methionine amino acid. So tRNA that reaches to the first codon or initiator codon called initiator tRNA complex. Now first step completed, that is activation of amino acid and transfer to the tRNA. It required enzyme amino acyl synthetase, energy in the form of ATP. Next step, initiation of polypeptide chain. Second step, initiation of polypeptide chain. In this one, initiating factors are involved. Initiating factor 1, 2, 3. 2 subunits of ribosome, mRNA, energy in the form of GTP involved in this formation of initiation complex. That is called initiation of polypeptide chain. Now come to this one. Smaller subunit of ribosome, that is the 30S, combines with initiating factor 3. It is initiating factor 3. It is the first step in this initiation of polypeptide chain. Next, smaller subunit with initiating factor 3 combines with initiating factor 1. Now the smaller subunit consists initiating factor 1, initiating factor 3. Meanwhile, initiating factor 2 that combines with GTP, that consists initiating factor 2 with the GTP. Next, smaller subunit with initiating factor 1, initiating factor 3 combines with initiating factor 2 along with GTP. Initiating factor 1, 2, initiating factor 3 with GTP. Now this combines with mRNA. It is mRNA, and this complex attached to the 5' end. So codon AUG, CCC for example. GTP hydrolyzes, all these three factors are released. Smaller subunit first attached to the mRNA at the 5' end region. Sequence again, first smaller subunit, that is combines with initiating factor 3. Next it combines with initiating factor 1. Meanwhile, initiating factor 2 combines with GTP, and it also combines with the smaller subunit with initiating factor 1 and 3. Now the whole complex combines with the mRNA, GTP hydrolyzes, all the three factors, initiating factor 1, 2, 3, all are released. Next step, tRNA with UAC anticodon that carry methionine reaches to the first codon. Hydrogen bonds are formed, weak hydrogen bonds, and then larger subunit attaches. After that, larger subunit attaches. Now this is called initiation complex. For the formation of initiation complex required 2 subunits of ribosome, mRNA, initiating factors 1, 2, 3, energy in this form of GTP, all are required. Initiation complex is formed. Now, first tRNA or initiated tRNA reaches to the P site. Now, A site is empty. A site is empty. So, larger subunit of ribosome contains 2 sites, A site and P site. Just listen. Smaller subunit, larger subunit. Consider 2 sites, P site, A site. P site also called polymerization site. Or donor site. A site acylation site or acceptor site. Only initiated tRNA reaches to the P site, remaining all tRNAs reaches to the A site. Except initiated tRNA, remaining all tRNAs reaches to the A site. Now, initiation complex torn. So, second step also completed. Initiation of polypeptide chain. In this, initiation complex is formed. Third step, elongation of polypeptide chain. So, in the second step, initiation of polypeptide chain completed. Next, third step, elongation of polypeptide chain. Now, another tRNA that carries second amino acid. Another tRNA that carries second amino acid. For example, second codon is the CCC, anticodon is the GGG. it carry the second amino acid to the A site. For this transportation, required energy in the form of GTP and a factor that is required, that is elongation factor T. To carry amino acid from cytosol to protein synthetic area, tRNA required two structures, one that is elongation factor T and energy in the source of GTP. It reaches to the A site and binds with the codon, anticodon, codon, both bind with the help of hydrogen bonds. Now, after attaching this one, tRNA in the A site, peptide bond is formed in between the first amino acid and the second amino acid. After the formation of the peptide bond, tRNA present in the P site released into the cytoplasm. After the formation of the peptide bond in between the first and the second amino acids, tRNA present in the P site released into the cytoplasm. Now P site is empty. Now, ribosome that moves to the next codon. The movement of ribosome to the next codon, in which direction it moves? 5 to 3 end direction. Also called CN direction. Why we call it the CN direction? So 5 to that means it moves 5 to 3 end direction, that is okay. And why it is called the CN direction? Peptide bond is formed in between the carboxyl group of first amino acid and amine group of next amino acid. Carboxyl group C is the important, amine group is the nitrogen part, so in direction CN direction. The movement of ribosome on mRNA in 5 to 3 end direction, 5 prime end to 3 prime end direction, or CN direction called translocation. Movement of ribosome on mRNA in the direction of 5 to 3 end direction or CN direction, translocation. Required energy in the form of GTP and factor elongation factor G. Here also two contents required, energy in the form of GTP and factor elongation factor G required. Now, due to its movement on the mRNA to the next codon, again A site is empty, P site contains the tRNA. That means the tRNA present in the A site change it to the P site. Actually, tRNA not moves, ribosome only moves. Because of the movement of the ribosome, the position of tRNA changes. tRNA present in A site become P site. Now A site is empty. Another tRNA carry third amino acid, reaches to this one. Bond is formed in between the second and third one. So it is continued up to the nonsense codon or chain terminated codon reaches to the A site. Once nonsense codon or chain terminated codon reaches to the A site, this polypeptide chain elongation stops. That gives the stop signal. This stop signal recognized by three factors: releasing factor 1, releasing factor 2, releasing factor 3. Nonsense codons or chain termination position recognized by three factors, namely releasing factor 1, 2 and 3. After that, No tRNA reaches to the A site. Peptidyl transferase that breaks bond in between the amino acid and the tRNA. So polypeptide chain released in the cytoplasm. Next, tRNA in the P site released into the cytoplasm. The two subunits of ribosome separate from one another, and then mRNA hydrolyzes into nucleotides. So complex completely separated. Complex completely separated. mRNA hydrolyzes into that is separated into the nucleotides. Two subunits separated. Polypeptide chain released into the cytoplasm. tRNA again released into the cytoplasm. It is called termination of polypeptide chain. This is the fourth step. Now, it is the process of the translation or synthesis of the polypeptide chain or synthesis of protein. It occurs in the cytoplasm. Requirements all these energy in the form of ATP for the activation of amino acid, and for remaining all the steps required GTP. Number of ATP required, that is equal to number of amino acids. Number of GTP required, that is equal to n minus 1. N is equal to number of amino acids. Number of GTP required in translation, that is equal to 2n minus 1. N is equal to number of amino acids. For example, in a polypeptide chain, 100 amino acids present. Number of ATP required, 100. Number of GTP required, 2n minus 1. That is the 200 minus 1. That is 199. Why it is 199? Reason, for the second step, for the formation of initiation complex, 1 GTP that is required. Out of 100, 99 reaches to the A site. So 99 required, 99 tRNA is required, 99 GTP. 99 translocations occur. So required 99. So 99 plus 99. So in the second step, 1, third step and fourth steps required 99 plus 99. Total 199. For initiation complex, 1 GTP. For each translocation, 1 GTP. 99 translocations, so 99 GTP. 99 tRNA reaches to the A site, each one required 1 GTP, so 99. 99 plus 99 plus 1, 199 GTP, that is 2n minus 1. Number of polypeptide chains equal to. This translation completed. Now, calculations. Number of peptide bond equal to number of amino acids minus 1. Number of codons minus 2. That is equal to number of water molecules release, that is equal to number of translocations, that is equal to number of translocations. For example, a polypeptide chain consists 100 amino acids. Number of peptide bonds. Number of peptide bonds. 99. Number of codons. Number of peptide bonds. Sorry. Number of peptide bonds, that is the 99. Number of amino acids. Number of peptide bond. Number of amino acids minus 1. 100 minus 1, 99. Number of codons. Number of sense codons, 100. Number of nonsense codon, 1. That is the 101. So, number of codons minus 2. That is 10 minus 2. That is 99. Number of water molecules released during the formation of each peptide bond, 1 water molecule release. So, it is also 99. And number of translocations, that is also 99. All are similar. Number of peptide bonds is equal to number of amino acids minus 1, that is equal to number of codons minus 2, that is equal to number of water molecule release, that is equal to number of translocations of ribosome on mRNA. That's all about the translation.